Hello everyone and welcome back to Dr. Wasif Academy. Today we are moving from theory into practical. As we have spent our recent sessions breaking down the structure and function of carbohydrates, fats, proteins, DNA and water, now it is time to see how these molecules actually behave in the lab. So in this session, I am going to walk you through the essential food test you need to master. We will cover the specific chemical indicators used to identify starch, reducing sugars, proteins, fats, and vitamin C. As a quick reminder, these practicals are not just interesting, they are a mandatory part of your IGCSE Biology syllabus. Understanding the how and why behind the color changes of various tests, this is vital for both your paper 3 and paper 6 exams. So grab your lab notebooks and let's get started. Now we move from theory into the laboratory. Okay, how do we actually prove these molecules are present in the food that we eat? We will start with the most famous practical, the iodine test for starch. Starch is a massive insoluble polymer of glucose and it has a very specific coiled spiral shape. This shape is the key to the test. When we add iodine solution, which is naturally an orange-brown color, those iodine atoms get physically trapped inside the center of the starch spiral. And this changes how the molecules reflect light, creating a distinct chemical reaction. The procedure is very simple. Place your food sample on a white tile or in a test tube and add 3 to 4 drops of iodine. If starch is present, you will see an immediate shift from orange-brown to blue-black. Remember this shift. If there is starch, then color will be shifted from orange-brown to blue-black. So if the sample stays orange-brown, it means there is no starch. Here you can see that a positive result will be considered if the solution turns from orange brown to blue black, and negative result will be considered if the solution remains orange brown. Why remain orange brown? Because orange brown is the natural color. Okay, so this is a marking scheme trap. On your exam, do not just write blue or black. Okay, to get the mark you must use the hyphenated term blue dash black. Okay, like this one, blue dash black. Also remember the safety protocol. Iodine is a messy stainer. Always mention wearing a lab coat and goggles to keep your clothes and skin safe. Next, we are testing for reducing sugars like glucose and maltose. These sugars have a special chemical superpower. They can reduce the copper sulfate in our reagent, changing its oxidation state and, more importantly, its color. The procedure for this test has one rule which you cannot forget, that is the heat rule. Okay, unlike the iodine test, this test would not work at room temperature. You must add an equal volume of below Benedict solution to your sample and then place it in a boiling water bath at least 80 degrees Celsius for 5 minutes. Okay, so what is the safety tip here? Never heat the test tube directly. Okay, uh, oh, the liquid, if you heat it directly, the liquid can spit or bubble over. Always use a water bath. What makes this test unique is that it is semi-quantitative. That is a key exam term. It means that color does not just tell us if sugar is there, it estimates how much is there. If in the result we have a blue color, it means there is no sugar. Okay, blue means zero. Green or yellow, it suggests traces or moderate amount of sugar. But if we have brick red color, it indicates a high concentration. So if you see a solid powder settling at the bottom of a red tube, do not worry. That is just a precipitate of copper oxide. So Dear students, watch out for sucrose. Okay, the table which I have shown in this slide, it is a sucrose. What is sucrose? That is a non-reducing sugar. If you put it in a Benedict, it stays below. So to test for sucrose, you must have to cheat by boiling it with dilute hydrochloric acid to break it down into glucose and fructose. So only then will be the Benedict test will work. As we have tested for energy, now let's test for the machinery of the cell. The Biuret test. Biuret test is our go-to method for detecting proteins. But here is a technical detail that students should keep in mind. This test does not actually detect amino acids themselves. It detects the peptide bonds that hold them together. This means you need a polypeptide or a full protein to get a strong reaction. The procedure is a two-step chemical process. First, we add an equal volume of sodium hydroxide NaOH to our food sample. This makes the environment alkaline, which is essential for the chemistry to work. Then we add a few drops of blue copper sulfate. So as you shake the tube gently, watch for a color shift. If proteins are present, the solution will change from blue to purple. From blue to purple. Okay, so you might also see it described in textbooks as violet or mauve. If it stays blue, the machinery is not there. So what is the science behind this color shift? Why does it turn purple? It's all about those copper ions. In that alkaline environment which we created, the copper ions form a complex with the nitrogen atoms in the peptide bonds. And this complex changes how light is reflected, shifting the color from a simple blue to that royal purple. So, my dear students, patience is a virtue in the lab. Okay, remember this term. Patience is a virtue in the lab. Unlike the iodine test, which is instant, the Biuret test can take a minute or two to fully develop. So, if you see a faint pinkish purple, it usually indicates a shorter chain of amino acids, which is often called as peptones. Finally, we come to the ethanol emulsion test. Lipids are non-polar, which means they are water-fearing and would not dissolve in it. Why water-fearing? What is water-fearing? We have also another name of water-fearing. If you know that, comment that in the comments section. So, no doubt that lipids are water-fearing and would not dissolve in it. They dissolve perfectly in ethanol. Means lipids dissolve in ethanol. So we use this property to trap the fat and force it to show itself. There is one golden rule for this experiment, that is the dry tube rule. So if your first test tube has even a single drop of water in it, the test is ruined before you start. You must place your food sample in a bone dry tube. Then you will add 2 cubic centimeters of ethanol, okay, and shake vigorously. It will dissolve any hidden fats. The second step is where the magic happens. You pour that clear ethanol liquid into a second tube filled with cold, okay, filled with cold distilled water. If fats are present, a cloudy white emulsion will immediately form at the top. Okay, here you can see this is cloudy white emulsion. If it is formed looking a lot like watered down milk. Okay, so if fats are present, the liquid stays as clear as glass. Okay, but why does it turn white? So because when the ethanol hits the water, the alcohol immediately dissolves into the water. But the fact, but the fat that cannot dissolve, it gets stranded in millions of tiny droplets, and these droplets are so small that they stay suspended, scattering light in every direction. That scattered light is what our eyes see as a white cloud. As a takeaway tip for your exam, an emulsion is a mixture of two liquids, that do not normally mix, like oil and water. So when you add the fat ethanol mixture to water, the ethanol dissolves in the water, leaving the tiny fat droplets stranded. These millions of tiny droplets scatter light, which is why it looks white and cloudy. Secondly, dry tube warning. Okay, if the first test tube has even a drop of water, I am repeating it, that if the first test tube has even a drop of water in it, the fat would not dissolve in the ethanol properly and the test will fail. As we have tested for the big four, but what about the micronutrients? For example, for vitamin C, which is also known as ascorbic acid. For this, we use a blue dye uh, called DCPIP. Okay, DCPIP. Vitamin C is a powerful antioxidant, which means it has the chemical strength to reduce the DCPIP, stripping away its blue color and turning it completely colorless. This is a titration style test. You did not just dump the juice in, okay? You add it drop by drop. You start with a fixed volume of below DCPIP and add the juice until the blue disappears. Blue color disappears, okay? Now listen closely, because this is where students often get confused. The results are inversely proportional. If it only takes 2 drops to clear the blue, if it takes only 2 drops to clear the blue, that juice is a powerhouse. It is highly concentrated. But if it takes 20 drops, that juice is weak. It has a low concentration of vitamin C. So the fewer drops you need, the stronger the vitamin C. If your practical exam, they will look for your fair test variables. You must mention using the same volume of DCPIP and the same size dropper for every sample. Also, if you are testing a dark juice like black currant, it can be tricky to see the endpoint. A pro tip for your answer is to suggest diluting the dark juice first, so the color change is actually visible. So if you only memorize one slide from this entire practical series, let it be this one. You can take a screenshot of this slide. This is your food test master table. Whether you are in a real lab or sitting for your exam, these 5 tests are the foundation of biological testing. Let's do a lightning round review of our positive results. Starch, use iodine. To identify starch, use iodine. If it is there, it will go blue black. If you are identifying, reducing sugar, use Benedict, and most importantly, heat. Okay, we are looking for that brick red precipitate. Brick red precipitate. If there is brick red precipitate, then the concentration is high. Then it's about proteins. For protein test, use Biuret, and sodium hydroxide and copper sulfate are used as chemicals. We are looking for a shift from blue to purple or violet. Then for testing fats, use the ethanol emulsion test. Look for that cloudy white emulsion at the top of their tube, and if you are identifying vitamin C, use DCPIP reagent. We are counting drops until the blue turns colorless. So take a screenshot of this slide, print it out and stick it on your wall. If you can recreate this table from memory, you have just secured a massive chunk of your practical exam marks. You have mastered the chemistry, the procedure and the results, and this is a great job. Before we wrap up, we need to talk about a common exam trap. What if your sample is not a liquid? What if you are asked to test a solid like a peanut or a piece of a nut? You can't just drop a whole nut into a test tube and expect the chemistry to work. You have to prepare it first. There are 3 steps you must include in your exam answer to earn full marks. Number 1 is grind and crush. Use a mortar and pestle to break the food into a fine mush. Okay, this increases the surface area, ensuring the chemicals can actually reach the molecules inside. Second one is extract. Add a small amount of distilled water or ethanol if you are testing for fats. Because if you're testing for fats, we'll have to add ethanol. So after adding water or adding ethanol, shake it well. This washes the molecules out of the solid and into the liquid. Then the step is filter. For this, use a funnel and filter paper to remove the solid debris. The resulting filtrate is the clear liquid you will actually use for your tests. So if you are asked why a sugar-free soda stayed below in a Benedict's test but turned red after you boiled it with acid, you have just found sucrose. The acid did the work of breaking that non-reducing sugar down into glucose. And if your fat test on that peanut fails, don't blame the chemistry. It is usually an experimental error. Either the nut was not crushed enough to release the oil, or your test tube was wet in step 1. So precision is everything. Now that you have understood how the test work, let's see if you can read the results like a scientist. This is extremely what you will see in an exam, a data table of unknown food samples. Let's decode it together. Look at the potato. It is the king of starch, turning that iodine blue black. But notice, it tests negative for everything else. But notice, but it's pure energy storage, okay? It is pure energy storage. Compare that to full fat milk. Compare this potato with this full fat milk. It's a biological cocktail. It tests negative for starch, okay, but it has a trace of sugar, a strong purple for protein, and that signature cloudy white emulsion for lipids. Then we have peanuts. Notice the purple and cloudy white results. Okay, purple and cloudy white results. This tells us peanuts are built for two things: structural proteins, okay, because purple is for protein, structural proteins and high density fat storage. Now look at bread. It is the most complex one here. Okay, it has starch, a trace of sugar, and notice that purple result. That is the gluten, which is protein found in wheat. So when you see a table like this in your exam, do not panic. Just look at the colors. They tell the story of exactly what that organism was storing when it was harvested. Do not confuse that I did not discuss about apple juice. You will discuss apple juice, okay, and you will give your response in the comment section, what it has, what it does not have. Okay, my dear viewers, I hope this session has helped you feel confident in performing and interpreting tests for starch, sugars, proteins, fats, and vitamin C. If any part of the procedure remains unclear, please drop your questions in the comments below. I make it a priority to answer them. Your feedback is essential in helping me refine these lectures to better meet your learning outcomes. If you have not joined us yet, hit the subscribe button and the bell icon to ensure you never miss a lesson. Best of luck with your studies, and I will see you in the next lecture. Goodbye.